Hi everyone, I am Veronica Reyes and I'm going to talk about a natural experiment in Sacred Fir, Ave religiosa, peri-urban forest affected by tropospheric ozone in Mexico City. Pollution is one of the strongest environmental changes that urban species face. One of the most common and harmful urban pollution is tropospheric ozone. In this heat map, you can see the daily concentration that Mexico City has been exposed to since the 90s. The color indicates the air quality according to the ozone concentration. Green is good and gradually worsens into purple. Although Mexico City control of air pollution has improved, it still exceeded what is established as tolerable, and during the dry hot period, days of good air quality in green are scarce. Mexico City is located in basin where the air current flow into the southwest of the city, and Sacred Fir forest at the southwest of Mexico City are the city's natural atmospheric drainage. Thus, these peri-urban forests are daily exposed to Mexico City's pollution, which in turn affects the vegetation. How ozone damage vegetation have been studied for more than 20 years in different parts of the world using field and laboratory experiments, as well as common controlled garden and growth chambers. It has been found that ozone enters plant cells through stomata and causes metabolic stress when it travels through the apoplast, producing different reactive oxygen species, ROS, and a particular color print in different species. In Ave religiosa, ozone damage include a reddish color on the needle's adaxial part and palisade parenchyma degradation in damaged needles. This color pattern differs from what can be seen in trees subject to drought or pathogens, where damage begins in the most apical part of the leaves. Despite relative improvements in air quality and reforestation attempts for decades, Mexico City forests have not recovered. However, in recent years, we observed phenotypic difference in ozone damage at the macroscopic level among young Sacred Fir individuals in forests heavily exposed to ozone. This variability could have a genetic basis and therefore lead to adaptation to ozone, either naturally or considering trees generation times boosted by reforestation programs. It is still needed to assess whether this variation is present in population threatened by ozone pollution in real settings like urban forest. Therefore, we collected some apparently tolerant trees as well as visibly damaged by ozone in Cruz de Colochtitla, the site of the greatest exposure to ozone. For our analysis, we collected needles from 3 growth periods and 2 ozone seasons, one moderately bad for human health and other really bad. We decided to explore this potential variability on ozone tolerance and the genetic basis with 3 questions. A. Macroscopic difference in natural setting correspond to histological and metabolic difference associated with ozone experiments? B. Is the difference between damaged and tolerant individuals related to their geographic origin? And C. Their genes differentially expressed between tolerant and damaged trees? Well, our results show that the individuals that we label as damage based on macroscopic characteristics indeed have symptoms of ozone damage at the histological level. This damage is different from other common sources of damage such as drought and diseases, which produce different symptoms both macroscopically and histologically. However, although our tolerant individuals are different from the damaged, we also found a slight cell collapse in some needles of tolerant trees. This could indicate that instead of two dichotomous states of tolerance to ozone, there could be a gradient. Metabolites detected in our samples have been reported in trees exposed to ozone. The production of metabolic compounds such as volatiles can remove rust before they cause severe cell damage. Our results are consistent with this, since we obtain higher concentration of metabolites in tolerant individuals compared to the damaged. For example, in tolerant individuals here, the tolerant individuals produced a graded quality of beta-pineno compared to those damaged in both periods. This increase in beta-pineno has been related to the stimulation of antioxidant mechanism induced by stress to ozone in Quercus plant. Furthermore, beta-pinene belongs to the group of monoterpenes which can promote resistance to ozone. In our results, the identified metabolites indicate difference between tolerant and damaged individuals in two periods of ozone exposure. We found genetic groups given by mountain origin. Detecting the origin of individuals in this type of study is important, given that different backgrounds have different degrees of sensitivity to ozone, and our study site has had multiple attempts at reforestation with diverse germoplants backgrounds. Cofre de Perote here and Nevado de Colima populations here have been reported with less sensitivity to ozone damage, while towns close to Mexico City and Nevado de Toluca have shown greater sensitivity. However, our analysis indicate that all individuals from Santa Rosa Xochiac or Cruz de Colochtitla are grouped with local population such as Ajusco and Nevado de Toluca, so our data indicate that both tolerant and damaged individuals seem to come from the same genetic group, and that they correspond to the center of the Trans-Mexican volcanic belt, without it being possible to determine a particular mountain of origin within this group. Most studies examining the genetic basis of ozone tolerance in plants have been performed in controlled experiments and in model organisms like crops. Here we contributed to identify differential expression genes under ozone exposure in a forest species in a natural setting with two methods, the SEC2 and HR, in pink here. And we have three comparisons. When Comparing damaged and tolerant individuals during high ozone exposure, we found similar differential expression in the transcript of the protein kinase, cytochrome P450, leucine-rich repeat, and legum-lectin domain to what has been found in controlled ozone exposure experiments in other species. When comparing periods with different levels of ozone concentration, we also found differential expressed transcripts that had been previously associated with ozone stress in the next transcripts. In this. And finally, we found transcripts that have not been related to ozone exposure in controlled sites or in settings exposed before, but we related with biotic stress in stress during plant microbial interactions and in crops and trees submitted to wounding or insects attack. In addition, some transcripts have been showed differential expression in response to abiotic stress treatments as temperature, heavy metals, salt, light, and drought. Our results are congruent with controlled experiments in other species, suggesting that plants' tolerance to ozone involves several mechanisms and pathways that also have other function in stress response. Most studies suggest that tolerance is specific to a species, but few studies have focused on existence of a tolerance gradient associated with genetic variation or its regulatory mechanism. Further, studies should focus on variants of these family proteins since they may be activating different tolerant response mechanisms inside the same species. So, we have some conclusion about this. The A conclusion is the first in our natural setting, exposure to ozone had similar tissue damage and metabolite production as controlled experiment with ozone concentration. B. Tolerant individuals could come from the Trans-Mexican Volcanic Belt Center, which is important to propose management plans and include local seeds in sites highly exposed to tropospheric ozone. And C. We identified genes differentially expressed between damaged and potentially tolerant trees similar in other plants exposed to ozone, and we propose other candidates to evaluate microbiome and environmental interactions. I would like to thank everyone who was part of this project and those who will continue work with us. You can ask me your questions here or now. Thanks.